Hello everyone. This lecture we are going to discuss the antibody. Just as the Dover Castle used walls and fortifications to defend their territory, the immune system constantly battles against foreign invaders such as viruses, bacteria, and parasites to defend the organism. Antibody molecules provide a key element in the immune system's defensive arsenal. For example, specific antibodies can bind to the molecules and the surface of the virus and prevent the virus from the infecting cells. The picture on the right is the antibody binds to one subunit of hemagglutinin from the surface of the influenza virus. Antibodies possess distinct antigen binding and effector units. Antibodies are central molecule player in the immune system. In 1959, Zuni Potter showed that immunoglobulin G, and it's also called IgG, the major antibodies in the serum, can be cleaved into three 50 kD fragments by the limited proteolytic action of the papain. The immunoglobulin G consists of two kinds of polypeptide chains, a 25 kD light chain and 50 kD heavy chain. The subunit composition shown in the figure is L2H2. Each light chain is linked to a heavy chain by a diphosphate bond, and the H chains are linked to each other by at least one diphosphate bond. Each L chain comprises two homogeneous domains termed immunoglobulin domains, and each heavy chain has four immunoglobulin domains. Overall, the molecule adopts a conformation that resembles the letter Y. The stem of this Y corresponding to the Fc fragment obtained by the cleaved with papain. In the figure, papain cleaves the H chains and the carboxy terminal side of the diphosphate band that link each light and heavy chains. Thus, each FAB consists of an entire H chain and the amino terminal half of a heavy chain, whereas FC consists of the carboxy terminal halves of both heavy chains. So each FAB contains a simple antigen binding site. It can be seen from the figure because an intact IgG molecules contains two FAB components and therefore has two binding sites. It can cross-link multiple antigens. As shown in the figure, the Fc and the two FAB units of the intact IgG are joined by flexible polypeptide regions that allow facile variation in the angle between the FAB units through a wide range. This kind of mobility, called segmentation flexibility, can enhance the formation of an antibody-antigen complex by enabling both recognition site and antibody to the bound an antigen that possesses multiple binding sites. The site at the tip of the FAB unit simply moves to match the distance between specific determinants on the antigen. Immunoglobulin G is the highest concentration antibody in serum, but there are other kinds of immunoglobulin, and the main immunoglobulins in the serum are listed in the table. As shown in the figure, each class of immunoglobulin includes a light chain, either alpha or gamma, and a distinct heavy chain. The heavy chain in the IgG are called chains, whereas those in immunoglobulin A or M or D and E are called alpha, delta, and sibling respectively. IgM is the first class of antibodies to appear in the serum after exposure to an antigen. The presence of the 10 antigens recognition site enables IgM to bind especially tightly to antigen containing multiple identical epitopes. IgA is a major class of antibodies in external secretions such as saliva, tears, bronchial mucus, intestinal mucus. Thus, IgA serves as the first line of defense against bacteria and viral antigens. The role of IgD has long been a mystery, but recent studies suggest IgD plays a role in the activation of basophils, white blood cells that have anti-parasite functions. IgE is important in conferring protection against parasites, but IgE also participates in allergic reactions. IgE antigen complex form cross-links with receptors and the surface of mast cells to trigger a cascade that leads to the release of granules containing pharmacologically active molecules. Histamine, one of the agents released, induces smooth muscle contraction and stimulates the secretion of mucus. Antibodies bind the specific molecule through hypervariable loops. The carboxyl terminal half of the light chain and the carboxyl terminal 3/4 of the heavy chains are very similar in all of the antibodies. Importantly, the amino terminal domain of each chain is more variable, including 3 stretches of approximately 7 to 12 amino acids within each chain that are hypervariable, as shown for heavy chain in this figure. The amino terminal immunoglobulin domains of each chain is thus referred to as the variable region, whereas the remaining immunoglobulin domains are much more similar in all antibodies and are referred to as constant region, as can be seen from this figure. An IgG molecule consists of a total of 12 immunoglobulin domains. These domains have many sequence features in common and adopt a common structure, the immunoglobulin fold and shown in the figure. Remarkably, the same structure domains is found in many other proteins that might play key roles in both immune and non-immune function. The immunoglobulin fold consists of a pair of beta sheets, each built of antiparallel beta strand that surround a central hydrophobic core. A single diphosphate band bridges the two sheets. Two aspects of this structure are particularly important for its function. So first, 3 loops present at one end of this structure form a potential binding surface. These loops contains the hypervariable sequence present in antibodies and in T-cell receptors. Variation of the amino acid sequence of these loops provides the major mechanism for the generation of the vastly diverse set of antibodies and the T-cell receptors expressed by the immune system. These loops are referred to as hypervariable loops or complementarity determining regions, called CDRs. Second, the amino acid terminal and the carboxyl terminal are at the opposite end of the structure, which allows structured domains to be stronger together to form the chains. And in the light chain and heavy chains of the antibodies, the immune globulin fold is one of the most prevalent domains encoded by the human genome. More than 750 genes encode proteins with at least one immunoglobulin fold, recognizable at the level of amino acid sequence. So for each class of antibody, the variable domains at the amino terminal ends of the light chain and heavy chain come together to form a binding surface. The position of the CDRs are striking. This hypervariable sequence, present in 3 loops of each domain, come together so that all 6 loops form a single surface at the end of each arm, as shown in this figure. The results of X-ray crystallographic studies of several hundred large and small antigens bound to the FAB molecules have been source of much insight into the structural basis of the antibody specificity. The binding of the antigen to antibodies is governed by the same principle that governs the binding of the substrate to the enzyme. The interaction between complementary shapes results in numerous contact between amino acids at the binding surface of both molecules. Many hydrogen bond electrostatic interactions and van der Waals interactions, reinforced by the hydrophobic interactions, combine to give specific and strong bonding. A well-studied case of small molecules bonding is seen in the example of a phosphoalkyl bond to FAB. Crystallographic analysis revealed phosphochlorine bond to a cavity line by residues from 5 CDRs to 4 from the light chain and 3 from the heavy chains. It can be seen from the figure. The positive charged trimethylamine group of phosphochlorine is buried inside the wedge-shaped cavity, while it interacts electrostatic with 2 negatively charged residues, a glutamate and the aspartate. The negatively charged phosphogroups of the phosphoalkyl bond to the positively charged guanine group of the arginine residue at the mouth of the crevice and is hydrogen bonded to the side chain of the nearby tyrosine residue. Numerous van der Waals interactions, such as those made by a tryptophan side chain, also stabilize this complex. The residues from 5 CDRs participate in the bonding of phosphochlorine to human FAB. This bonding does not significantly change the structure of the antibody, yet induced fit plays a role in the formation of many antibody-antigen complexes. A malleable binding site are accommodate many more kinds of ligand than can be rigid ones. How do large antigens interaction with antibodies? A large collection of the antibodies against hen-egg white lysosome has been structured characterized in great detail, as shown in this figure. So each different antibody binds to the distinct surface of the lysosome. Let us examine the interaction of the complex 2 in the previous slides. As shown in the right figure, this antibody binds to polypeptide segments of the lysosome that are widely separated in the primary structure. All 6 CDRs of the antibody make a contact to these epitopes. The region of the contact is quite extensive. The opposite surfaces are rather flat. The only exception is the side chain of the glutamine 121 of the lysosomes, which penetrates deeply into the antibody's binding site. Well, it forms a hydrogen bond with a main chain carbox oxygen atoms and is surrounded by 3 aromatic side chains. The formation of 12 hydrogen bonds and the numerous van der Waals interaction contributes to the high affinity of these antibody-antigen interactions. Here is some reference, and thanks for your attention.